Hello and welcome to another interview. This time we're with Dr. Deus Mugabe. Would you care to introduce yourself?
Oh, my name, as Brandon just put it, my name is Deus Mugabe. I'm a scientist or breeder with Bayer Crop Sciences. Currently, I'm working on breeding new corn varieties for the northeast region of North America. So that includes Ontario, Quebec, New York, Pennsylvania, and parts of Michigan as well. I finished my grad school PhD at University of Guelph in 2024, where I was working on identifying the genes that are responsible for white mold disease resistance in soybean that's grown in Canada. Before that, I completed a master's at Washington State University, where I worked mostly with post crops. Specifically, my master's looked at cold tolerance for chickpea. Identifying QTL for cold tolerance in chickpea. Before that, I grew up and did most of my studies before that in Rwanda before completing a bachelor's in crop sciences in the Philippines. I think that's mostly about the introduction.
Excellent. During your PhD project, you worked on trying to identify QTLs in soybean. Could you briefly explain what your project entailed and what the basic gist of what that project was?
Yeah. So basically, my thesis was the genomic analysis of Sclerotinia stem rot, also known as white mold, that is caused by a fungus called Sclerotinia sclerotiorum in soybean, in a panel of soybean that's grown in Canada. So basically, my project had a couple of objectives. But the main one was to identify the genomic regions that are associated with partial resistance to white mold in that panel of soybean that's grown in Canada by using genome-wide association studies. And
Ec-
then to follow up on that objective, I also had to do some cross-validation of the identified genomic regions using an independent subpanel of soybean. So this is a kind of like a separate subpanel of soybean that's also grown in Canada, but wasn't part of my main panel. Um,
ah,
yeah, and I can just also outline two more objectives that we did I did work on with my advisor and a couple other collaborators, even though we might not get enough time to go deeper into those. But they might be worth mentioning. So after cross-validating the identifying the cross-validating of the identified regions, genomic regions, we also worked on identifying potential candidate genes that are again responsible for partial resistance to white mold, and then also examine their involvement in the expression of the trait by using real-time gene expression analyses. And then lastly, we also did examine if there is any potential penalty that is associated with carrying partial resistance alleles on the agronomic and seed quality traits in that Canadian soybean panel.
excellent. Thank you. Based on your work, you utilize GWAS in order to find these genetic regions or QTLs.
Yes.
Could you explain what GWAS is and how it can be used to find these QTLs?
Yeah. See how I can put in a simple word. So basically, GWAS is one of the techniques or methods that is commonly used in plant breeding, and it's used to find genes that are linked to important traits, such as disease resistance, drought tolerance, or higher yield. So basically, scientists can collect a number of different genotypes and then screen them and screen the trait. So for example, in my case, I was looking at white mold, so we had to use a method to find variation in very many different genotypes, variation for that disease, and then as well take some genetic information. So just basically getting some DNA from these genotypes. And then once we've done this, we can kind of like do some mapping and see what genes or what genomic regions can explain the variation in that trait of interest, in my case, which was um, resistance to white mold disease.
Ah, excellent. What type of QTLs for white mold did you find, and could you trace these QTLs back to a specific parental genotype or genotypes?
Yeah, so in my studies, I did find two. I might need to dig into some of the. It's been a while since I looked at my thesis, but I do recall that I did find some. Again, we call them QTL or marker trait associations. I did find some marker trait associations on chromosome 2 and chromosome 9. Basically that kind of like explain the shorter lesions in the panel that we had. So if I can kind of like provide a little bit more context on that. So when I mentioned that you screen for variation for your trait of interest, in my case, because we're looking at that variation for white mold disease resistance, the way we quantified that was we used a method that's called cotton pad method. This is where we kind of like use a cotton pad and that's dipped into an inoculum. So inoculum contains the fungus that causes the disease. Put it on the flower because that's mostly naturally how the fungus infects the plant. And then after about 7 to 8 days, the plant will start to develop the symptoms. In our case, the symptom would be the lesion or the injury. So you could imagine that the lesion size could also be could explain the resistance. So for example, if a plant has a longer injury, that means that it's susceptible compared to like a plant that has a very short injury or doesn't have an injury, right?
Exactly.
So the QTLs that I've identified, one on chromosome 2 and another one on chromosome 9, were associated to shorter injury size in the panel. And in terms of being able to map them to the parents, so something that I can probably highlight is that there's different ways, and I know I'm kind of using some of these terms interchangeably, but scientifically it might be not accurate. So there's different ways of mapping genomic regions, right?
Hmm.
So QTL mapping is most times used in relation to mapping used what they call a biparental population, or in other words, this is a population that's developed from two parents that differ in a trait, right? So,
Hmm.
for example, if I did use a biparental mapping population for my for soybean disease, so white mold disease resistance, I would get like one parent that is very susceptible and another one that is uh, very resistant, and cross them and develop a number of recombinant inbred lines. So just kind of like inbreed this the offsprings for up to 5 or 6 generations. And then after that, you will be able to kind of like trace back the genomic regions that are more coming from the resistant parent or the or the susceptible parent. But in my case, I use the genome-wide association study, which does not it's more associated, or it's the term mostly used here is marker trait associations. So we don't use the parents per se. We use individual genotypes, very many different diverse genotypes, right? So and this way we can use this method called genome-wide association studies, and you can find the genomic region that explains the trait of interest in all these diverse genotypes, if that makes sense.
That makes sense. So basically, you found a way to create little markers for different portions of the genome to
Mhm.
see if that portion of the genome is present or absent within a particular soybean line to determine if it's resistant to white mold or not. Is that
Ye-
correct?
Yes, yes. So yeah, so that yeah, part of the genome, if it explains resistance or susceptibility in all the different genotypes that you have in a panel, right? I
Right.
think, yeah.
Yeah. Thank you.
Of course.
Uh, so any specific conclusions you came to as the result of this project? Anything interesting that showed up that you didn't expect to see?
Um, well, I guess the main, say, big result there again was the two marker trait associations that we found, one on chromosome 2 and another on chromosome 9, that I believe both of them explained about 30% reduction in injury size,
Wow.
right? So which if you know if that would be validated even further and confirmed, that would mean it could have big implications or impact in terms of like reducing um susceptibility to that disease in soybean, right? So I
Exactly.
think that's a major result, but also it was pretty interesting um when we did the validation using a separate sub panel. And I'm not gonna be able to recall 100%, but I do remember that the validation study corroborated what we found in the GWA study. So basically, even within an independent small panel, we found that the alleles that were explaining resistance in the bigger panel also did explain resistance in that smaller subpanel, if that makes sense, which kind of like is consistent, right?
Exactly.
Mhm.
Thank you. And since we have used up all the questions, I'll finish this off by asking you what is one thing that's not being discussed in agriculture that should be discussed a lot more?
One thing that's not discussed in agriculture that should be discussed more.
Yeah, what do you think should be discussed more in agriculture that's not being discussed right now?
I would say that I think, and I, I don't know if it's not discussed at all or if it's not discussed enough, in my opinion, but if it would be classified as not being discussed or just not being discussed enough, is that I think agriculture is getting to, so historically, agriculture is known to be more like a very manual, labor-intensive job that or field that, especially younger generations are not very attracted to. They don't, maybe, they don't find it very attractive or like a good career path to take. But we're seeing or we're entering a time, and we've been kind of like going this direction for some time now, where we have a lot of groundbreaking technologies that are simplifying it and allowing us to kind of um, um, hit all main major, I guess, like goals or objectives in agriculture fast and very efficiently. So to be more specific, there is that a lot of times we find that we want to maybe improve uh, production, right? Or we want to or we want to kind of like be more efficient with the resources that we use, or a number of things that we'd like to like do in agriculture. And now we have lots of cool technologies and tools that can help us solve this scientifically. And now we're also entering the era of AI where we can, and big data, where we can collect this massive amount of data and process it very, very, very fast and use AI and highly complex computational biology tools to kind of like extrapolate and get like really more accurate results and fast. So I guess what I'm kind of trying to drive to is that I think this is getting the field of agriculture is getting more and more and more exciting and should probably be discussed more in terms of like attracting the younger generations to get in there, because you find that one of the challenges is you find that a lot of young generations leave the field just to go and find I guess more exciting and attractive fields in the cities, while actually agriculture is looking like it's getting more exciting itself. So yeah, that's what I would say.
Thank you. I think that should cover everything.
Mm-hmm.
It's been a pleasure.
Yeah, thank you so much for chatting with me.
If you want to donate to my channel, the links are below. If you want to buy my book, you can also find that link in the description box below.